Okay, very good. So welcome everyone to the Precision Medicine seminar series. It is my pleasure to welcome our guest speaker today, Ben Neale, who's an associate professor in medicine at Harvard Medical School. He co-directs the program in medical and population genetics at the Broad Institute. He is the director of genetics at the Stanley Center for Psychiatric Research, and he is the director of genomics of public health initiative at MGH. Dr. Neale studied genetics at University of Chicago and Virginia Commonwealth University, and then he moved on to London to King's College, where he worked with Pak Shan on his PhD. He earned his PhD over there, and then he was recruited by Mark Daly to join his team at the Broad Institute. And he has a broad array of interests. He's very much interested in genetic causes of complex human traits, and he's developing new methods, statistical methods for the analysis of human genetic data with various applications, with a special focus on neuropsychiatric disease. He is also very collaborative and involved in multiple consortia, including the Brainstorm Consortium, as well as the Psychiatric Genomics Consortium, where he's leading a lot of analytical and sequencing work groups. He's also part of the International Common Disease Alliance, where he co-chairs the MAPS work group. And his lab developed a variety of different methods that I think everyone here is familiar with, at least some of them. More recently, LD score regression for genome-wide genetic correlation analysis, and HALE for as a platform for kind of big data analysis, and many more, but I'll let him speak. So welcome, Ben, and thanks for visiting us.
And thank you for the invitation. It's really a pleasure to be here. Nice to be in person. I've had a lovely time here at Columbia, and I'm going to talk to you today about genetic analysis at scale. Depending on how we go, I might tell you some things about the mitochondria at the end, but it's precision medicine seminar series, so I'll focus more on human health outcomes. Before I get started, I'd just like to do a few disclosures. So I serve on a variety of scientific advisory boards and have equity in some biotech startups. And you can see the disclosures there. All right, so this is sort of where we are today in human genetics. I think the theory of the case, if you will, is we're trying to go from samples, collecting genetic variation on those samples, associating them with phenotype, and then trying to understand the genes, the cell types, the cellular processes, the biological networks that give rise to shaping human complex outcomes. From schizophrenia to diabetes, genetics plays some role, is some source of risk, but it is not, the only source of risk out there. Obviously, the environment matters hugely. But I think there are lots of questions that the last, say, 15 or so years of genetic investigation have raised, and included amongst those are, what are the variants that really matter? We can find GWAS loci. How are we doing at fine mapping? How do we think about the consequences of that genetic variation? And how do we just go about this business of interpreting genetic associations to get at disease mechanism for hopefully novel therapeutic hypotheses, maybe patient stratification, maybe the identification of biomarkers, all of the various aspects that might facilitate biomedical research writ large. And I'm not actually going to answer many of these questions because I think they're still very much open. But I think it is very important to at least keep some of the sort of long-term goals of the activity and enterprise in mind when, we get into the weeds of how it all works and what we might be learning and how we might be proceeding. And to discuss this, I think there's been a number of large-scale biobanks that have been generated and are starting to or successfully sharing data. UK Biobank's a kind of great, perhaps even marquee example, where there are 500,000 individuals recruited across the UK. I always like to show the age of recruitment figure because there's a big bump over here. You can almost see this like increasing recruitment rate over age. And then there's this big jump, and there's like age of retirement effects. So retirees are way more likely to be participating in this cohort than people that are working. And that is just apparent, just looking at age, The other thing that is quite apparent in UK Biobank is the healthy volunteer effect. There's also the fact that women tend to engage in research activities more than men. This has been shown many, many, many times in many, many different contexts, and UK Biobank is no different. There's certainly this healthy volunteer phenomenon, so it's not perfectly representative of the UK population writ large. 5-year mortality is lower, rates of smoking are lower. There are a whole bunch of things that are subtly different on average amongst individuals in the UK Biobank than in the, say, UK population writ large. But those differences are, modest, and there are some methods and approaches to try and accommodate those differences as well. Now, UK Biobank is fantastic for a variety of reasons. I'll talk about our systematic analyses of it a little bit later, but it's also, I think, perhaps the most useful testbed for statistical methodology as it pertains to genetics that is out there today. And almost every statistical genetics paper that you read currently will have some empirical application to UK Biobank. And that is a reminder that the value of data sharing goes beyond just the understanding of the phenotypes in questions. It also serves to enrich and enable method development, new ways of asking questions about how people get sick or how they develop diabetes or whatever. All of those things are made possible by data sharing. So I'm a shameless advocate for data sharing. I think the New England Journal of Medicine might call me a data parasite, but I embrace my parasitic identity, and I would call for even more aggressive data sharing than we see today. Now, let's talk about fine mapping. So some nice slides from a former PhD student with Hilary Finucane and Jacob Ulrich that I think distills exactly what we're trying to accomplish when we find a genome-wide significant locus. Okay, so here the notion is we've done some association, we're correlating common genetic variation with some disease or phenotypic outcome, and we get this sort of locus zoom plot that you can see over here that has the sort of strength of association in the y-axis minus log p-value, and then the position on the chromosome on the x-axis. And the goal of fine mapping at its core is to turn these kinds of associations where LD contaminates the pattern of association and therefore makes it very difficult to tell exactly what SNPs are driving that locus. So fine mapping is trying to basically unwind that. It's trying to turn these genetic associations into a much smaller set of variants that are really the source of that association signal. And you have to juggle the LD and go through that sort of thing. The main readout for these kinds of approaches are often posterior inclusion probabilities. A lot of these methods are built on Bayesian rather than frequentist statistics. But these PIPs are really just a reflection of the chance that the algorithm thinks that this SNP is actually responsible for the association that you see. That's the goal of fine mapping. The other thing that people often do is to find some credible set. Credible set's often maybe a 95% or maybe 99%. And the idea here is that you sum up all of the posterior inclusion probabilities of the SNPs in the locus to give you the smallest set of variants for a single effect together that will have greater than a 95% chance of truly containing that causal variant. Okay, so we've got these posterior probabilities. We can think of a set of them. We're trying to get those sets down. And the reasons for fine mapping are many and varied. But I think some very natural and obvious ones to flag are, if I wanted to do something like base editing of those mutations to probe the regulatory consequence, I do not want to have to do every single SNP in this region. That's a much, much more expensive experiment than if I can distill down into, say, a set of 5 or 10 or 20 candidate variants out of the hundreds of variants that are often in the kind of shadow of the LD of the association. The precision by which you can fine map also may help figure out what the biological consequence really is. So just as a hypothetical, you might have an enhancer that is present in one cell type, next door a different enhancer that is present in a different cell type, and where that SNP is may relate to whether or not it's actually falling in the enhancer or outside of it. Whether it's inside or outside of the enhancer might be relevant to its function. And as we start to try and learn the rules of gene regulation, which we're still early in our tutelage, we're in like kindergarten, maybe nursery school of gene regulation rules, but still, there's a little bit of progress, some ideas out there in the community and the literature. Being able to know what SNP is driving the association may facilitate identifying what gene is being perturbed, in what cell type or what context that perturbation is happening. And that will give you a much more concrete starting point for developing a biological mechanistic hypothesis for how that association is translating into disease risk. So super important. Now, whenever these kinds of methods come out, there are a bunch of simulations based on usually simulated data, completely fabricated data, that will give some guidance about how well calibrated these posterior inclusion probabilities. And for pretty much all of the fine mapping methods, doesn't matter if you want SUSY or fine map or take your pick, pretty much all of them are well calibrated in simulated, completely fabricated data, where everything works like the model says it should work. What happens in real data? So that's the question that Ran Kui and Hilary Finucane took on. There's a preprint that you can see down there from bioRxiv. So take UK Biobank, you have this large, well-powered genome-wide association study. You do a GWAS, but you don't do a GWAS on the entire cohort. No, you downsample to 100,000 individuals and do that GWAS. And because UK Biobank's so big, you still find plenty of associations for plenty of outcomes. And so you have this little Manhattan plot over here showing every SNP chromosome base pair position against minus log-10 p-value. You find some associations. While you grab those associations, you fine map those loci, right? So we've got the locus, same sort of thing. We run some fine mapping software, SUSY, fine map, whatever, it doesn't matter. And then do it all again. But instead of doing it all again on the same 100,000 individuals, you can move to a new set of 100,000 individuals and rerun the entire edifice and enterprise and see whether you rediscover the locus. If you do rediscover the locus, what does the fine mapping look like? And that gives rise to a very natural sort of measure of the accuracy of these approaches, which is what Hilary and Ren describe as the replication failure rate. So the notion behind the replication failure rate is if you have a SNP that comes out of a single GWAS and has a 99% posterior inclusion probability, that means, according to the algorithm, that it's 99% chance of being causal for that phenotype. So then you go and do a separate independent GWAS. Again, remember, nice thing about UK Biobank, ascertained the same way, mostly genotyped on the same platform, like almost completely genotyped on the same platform, processed together, large data set, good principal components, high quality analysis. You have a lot of trust in the underlying GWAS. It's not like some meta-analysis where you're trying to integrate across lots of different structured data sets that might have different patterns of missingness and all that kind of stuff. No, this is all very homogeneous as these kinds of studies go. And so you have that 99% posterior inclusion probability of a SNP, and then you would expect that 99 times out of 100, when you have a SNP like that, it should show up in the second time that you do it. And that's where the baseline expectation is calibrated. Here's our ideal simulation. So not even a simulation from the authors, simulation from Hilary and Ran and colleagues. And yeah, methods are pretty well calibrated. In the UK Biobank data, we're seeing a much higher than expected replication failure rate. So that is to say that the high confidence posterior inclusion probability SNPs from UK Biobank from 100,000 individuals do not always turn up in subsequent analyses. Indeed, we lose maybe 10 or 15% of those SNPs in subsequent analyses. And you might say, well, maybe this is something about traits. Well, you look across a variety of different traits and you see, broadly speaking, exactly the same pattern, where, even for things like height or bone mineral density, take your pick of phenotypes. You have simpler genetic architectures like LDL or ALP or hemoglobin A1C, all of these different phenotypes are showing exactly the same phenomenon. It's kind of hard. Fine mapping is hard. Don't trust the algorithms. Lesson 1. So Hilary and Ran, not content to just leave it there, hit on the idea of maybe using some infinitesimal local effects. So if you just allow for a little bit of infinitesimal signal across all of the SNPs in the region, what does that do? Well, it does improve the replication failure rate, doing a good job, but it's still very far away from where we would calibrate the expectation based on the performance of the algorithms. There are a lot of possible explanations for this. I don't have an answer for why it's happening. Potential explanations include things like there are other forms of genetic variation that are not perfectly characterized by the SNPs. So if like some sort of STR or indel or other kind of genetic variant in that locus or region is really driving the association signal, the SNPs will still pick it up because population genetics is true for all classes of genetic variation, not just SNPs. So LD will be there, it will pull things through. And so you will never actually get a consistent fine mapping answer in that circumstance. If it's driven by some kind of variant that we're not looking at, that's a possibility. It could be that there are non-additive local effects. So all of these models, fine map Susie, fine map Bank Susie, and whatever, take your pick, they fit additivity at the sort of genetic effect level. And so it's like, pick the SNP with the biggest additive effect, go to the next one, et cetera, et cetera. If there are haplotype effects, if there is some non-additivity in the combination of multiple associated variants in that region, that could potentially be a violation of the assumptions of the model, lead to this kind of behavior. There could be, yep, go ahead.
My question is like, you said a threshold, like say 99%. Like, what about the ranking? Like, do you still pick the top? If you just pick the top, you pick
I love the hope. I love the hope that springs eternal from that question. If the like top high confidence ones are, there are cases where it's like 99% posterior inclusion probability for variant, and then it's like 0.01 in the next go round in terms of that chance of being the true causal SNP in that locus. So it is not simply that where the ranking is calibrated is appropriate. There is reordering of SNPs within a locus quite regularly in this kind of exercise. The other, I guess, thing that I would say in terms of, what might be going on are that, so we've got non-additivity, we've got lurking confounding biases, and then we've got genetic variation that we're not looking at. You might think that adding in functional annotation information could get you out of this well a little bit. And we looked at exactly that. And when you do that, here's the sort of background rate of the different, it's just a tripartite classing of genetic variations, so coding variants, putative regulatory variants, those are things that are falling in enhancers, that sort of broad view of gene regulation being incorporated. And then most of the other variation is non-genic. And here are the 700 replicated fine mapped associations where there is consistent posterior inclusion probabilities across subsets of the data set. You can see that they're quite strongly enriched for coding variants and for putative regulatory effects. So that gives us some confidence that, okay, maybe functional information can help you and it can make it a little bit more reproducible. One second. But even amongst the non-replicating SNPs, so the things that went from very high PIP to very low PIP in subsequent data set analyses, there you see a slight enrichment of coding variants against baseline. And that's because GWAS loci are not randomly distributed across the genome. They tend to occur around genes. When you tend to occur around genes, the algorithms will think that it's more interesting, that it's going to be, that it's just the stuff that's in physical proximity is going to have more LD. And so even functional information cannot rescue you with certainty about exactly what the SNP association looks like.
Just one potential reason for non-replication I just wanted you to comment is perhaps, you're sampling the same sample size for both cohorts of 100, but there might be still, big differences in terms of the distributions, right? So there should be a power differential. So have you thought about like making the replication cohort much larger than the initial cohort?
That's an interesting question. Yeah, so the question was sample size representation. In the quantitative trait case, it's largely moot because it's all randomly selected from across the distribution, and so it really isn't going to make much of a difference. There is another potential explanation that I failed to note, and that's some phenotypic heterogeneity. Now, I mean, like for things like height, I kind of have a hard time believing that we're measuring something different in a group A people versus group B people when we're just measuring height. But for binary outcomes or anything where there's some heterogeneity in what is actually being measured at any underlying level, that can also drive this kind of phenomenon. But for the quantitative traits, I ascribe a pretty low chance of that being the reason. Okay, any other questions about finding that?
But in theory, that heterogeneity is done by seeing a different distribution of GWAS signals. Right?
It's true. It's true. So the fact that you're finding the same GWAS signal, and we only look at the same GWAS signals,
Okay.
does certainly diminish the chance that this is being driven by heterogeneity, but it's still, I suppose, technically a possibility.
And those SNPs, so you have one SNP with a PIP of 99%. So that SNP means that it's not an LD with other SNPs around.
Or, well, there's not an LD, and then there's sufficiently strong, let's go all the way back here. There's sufficiently strong association that it stands out from the crowd quite compellingly. So if this SNP up here were all the way up like where the dot of the eye is, or if there was another SNP with the p-value where the dot of the eye is, the algorithm is almost always going to favor that.
So my question would be, so for the next figure, so no, sorry, yeah, this one. So how define? So you have, so which means that you have two sets of the SNP to pick up. One is the original cryptostat, the other one is the replicated cryptostat. How do you define that? How do you choose that? So do you mean that you only choose the SNPs with a larger than 99% of PPP?
In the first one, and then look it up in the second.
That kind of can be problematic, because I mean, we just did a research doing fine mapping on Alzheimer's disease, and for 30 loci that we analyzed with global significance SNPs with result, besides the ApoE, because ApoE is the W. There's only one loci with the SNP with PIP larger than 99%.
Yeah, well, I think that GWAS for height in 100,000 individuals is likely better powered than the GWAS in Alzheimer's that you're talking about.
Okay, so whenever, because I mean, I have doing a fair share of fine mapping, so whenever I see SNPs with extremely high PIP, first instinct I wouldn't trust that actually, because I will consider, okay, maybe something's wrong, because I mean, oftentimes the SNPs always have some kind of high LD with, no matter how strong it is, if you have LD with high LD with other variants, all the PIP will reduce, right?
I agree.
So I guess it would be better to do this for all the tribal staff instead of just the high PIP.
Yeah, well, but if the individual SNP PIPs are miscalibrated, then the credible set has to be miscalibrated. There's no two ways around it, right?
Yeah.
And I mean, I take your point that maybe you're interested in how good is it if we allow ourselves a little bit more uncertainty or allow ourselves more SNPs. But I think, I guess the point still remains that if we have evidence that the PIPs that are used to compose the credible set are wrong, or at least potentially upwardly biased, which is, I think, the claim, then that'll pervade into that space.
Okay, one more point.
Sure.
So, because I mean, so actually, so this kind of credible set was first proposed in Susi's paper, especially for the multi-credible set situation. And the spirit of credible cells, philosophy of doing this credible set is that we don't want to focusing on one single SNP. We want to introduce the uncertainty because of the high LD we cannot distinguish the order. Because if you have a popular annotation, a totally different picture. So what I'm saying here is that for me, the credible set is more like, okay, you have 100 SNPs, and you somehow doing fine mapping reduced to 5 SNPs for your credible set, and those 5 SNPs would be valuable for the following experimental measurements. So that's what I'm saying, that maybe it would be better to focus on the credible set rather than just one SNP.
Yeah, I mean, but even if so, in the 99% credible set notion, if we had a 2-marker haplotype that was in perfect LD, it would be subject to exactly the same bias. I don't see how the bias goes away just because you're conditioning on a 2-marker haplotype versus a single SNP, unless you think that, yeah, I think the math bounds it that way. I don't think there's any other option, unfortunately for us all. Not an easy problem to solve. All right. let's talk about what we're going to do with biobanks worldwide for a second. So UK Biobank's a fantastic resource, really exciting. It's not the only biobank in the world. There are biobanks popping up everywhere. There's one here, I understand. And to sort of power systematic genetic discovery across many different human disease outcomes, it seems like the best path forward is to try and work together, right? Like that's worked very well in specific disease entities. And so with that sort of spirit of let's work together and bring the community together, a few years ago, we launched the Global Biobank Meta-Analysis Initiative with Kristen Willer, who's now at Regeneron, and Ida Sareka at Michigan has taken Kristen's roles and responsibilities in the context of this effort. But with Wei Zhu, the kind of very talented method developer and statistical geneticist, as the sort of point lead for the consortium, alongside Mark and then Sinead Chapman, who organized all the project management activities associated with this enterprise and endeavor. We've sort of tried to bring the world's biobanks to the best of our ability together. It's an open collaboration, all are welcome to join. We, set on a set of demonstration phenotypes that I'll kind of talk through. But the first raft of papers from GBMI were published in Cell Genomics, I think late last year, if I remember correctly. And just a few notes about them. So we took 14 common endpoints across 18 biobanks and meta-analyzed the GWAS and then added in an additional 5 for replication. The largest, I think, disease-oriented studies were things like asthma, and I'll talk a little bit about asthma here. But in addition to that, there's kind of good discovery in asthma, increased the number of loci identified up to nearly 180, 49 of which were not identified in the Translational Asthma Genetics Consortium. But I think in addition to this question of what variants matter, there's the question of what genes matter, right? I think maybe we don't need to know the variant. Maybe we can just content ourselves with identifying the gene that is being perturbed by the genetic variant, because that'll get us to the biology. And if your interest is the biology rather than the genome, so biology of the disease, then absolutely this is a sensible direction to push on. And so we made this plot, which took 5 different approaches for gene prioritization, including TWAS, DEPICT, the PWMR, which is the sort of MAR approach with proteins, so protein-wide Mendelian randomization, and then POPS, polygenic backbone-driven priority score system that Hilary Finucane developed, and then just the nearest gene. Nearest gene is always a nice thing to include. Geography is great. And this is the upset plot that we get. So each of these histograms are linked to the methods that are agreeing or not for the different gene prioritization. I see some titters in the audience and laughing about how inconsistent this upset plot is. And I think this is another core lesson about where we are right now, is that it is very easy to make a guess about what the gene is at a genome-wide significant locus. It's actually very difficult to get agreement across different approaches for gene prioritization for what the gene is. So healthy skepticism is encouraged when trying to address what genes matter. There's another way, though. Human genetics, hope springs eternal, analyzing exome coding variation. So here, what we've got represented is GeneBAS. So with Konrad Karczewski and many of the Broad, we ran a systematic exome-wide association analysis for many thousands of phenotypes across UK Biobank. We ran burden tests, SCAT-O tests, focusing on LOS, on missense mutations, combining them all together. GeneBAS is fully browsable. And here is an example sort of gene Manhattan plot on the left-hand side for a phenotype called chronotype. My favorite phenotype in the world right now. And we find a couple of exciting associations. So here all of the associations for all of these tests are laid out, as well as a sort of variant browser to dig into what the mutations are that live in the association tests that were being generated. We have some guidelines about the reliability and trustworthiness of the association analyses, because when case numbers get small and very imbalanced against the number of controls, the association test statistics get unstable, particularly when the number of rare variants gets rare, like few mutations in the test, quite unstable, quite worrying. So what's chronotype? What am I banging on about here? So chronotype is just defined this way. This is the question they ask in UK Biobank. Are you a morning person, more morning than evening, more evening than morning, or definitely an evening person? I sometimes like to ask the audience. So, morning people, raise your hand. Yeah. Evening people, raise your hand. Yeah. And who's in the middle? A few. Yeah, people are always a little more like, there's more people in the middle than there are morning or evenings. Maybe something about science. And definitely more evening people in this room than we see in the UK Biobank proportionally. Well, the top association that I showed you in GBAS is to PER2, so loss of function mutations, so point nonsense mutations, combined allele frequency, pretty rare, 2 times 10⁻⁴. But 77 different putative mutations collapse together into the association test, 5 times 10⁻12 for that burden association test of LOX mutations. Those of you that know things about circadian rhythm will know that PER2 and PER3 are essential components of the primary negative feedback loop of the circadian clock. And there's this really nice paper from Louis Pachek from, I think, something like 10, maybe 20 years ago now, that using Mendelian genetic approaches mapped exactly PER2 loss of function mutations and pedigrees where extreme early morning chronotype was the phenotype. Individuals would go to sleep at like 7:30 at night and then wake up at like 5 in the morning. And that was just the way their circadian rhythm operated. So biobanks can be like transformative for gene discovery. And the nice thing about the rare coding variants in particular is, I just told you a story about a gene. I have to do some dancing to tell you a story about a gene from a GWAS locus, unless maybe it like is really just one gene in the locus and there's nothing else out there in the big wide world. So with that spirit in mind and seeing the success of GBMI, we launched the BRAVA consortium to sort of try and bring together the world's sequence cohorts, recognizing that sequence data analysis, both on the bioinformatics, the quality control, and the association testing front, are all actually quite different from the same protocols and pipelines that we've developed for common variant analysis on the back of genotyping arrays. So in partnership with Cecilia Lindgren, Konrad Krajewski, and Duncan Palmer, we're trying to work together to find if we can systematically analyze human diseases now across biobanks with things like electronic health records to build the same sort of knowledge base that we hope to build on the common variant side in GBMI. Again, open community, all are welcome. If you're interested, feel free to reach out. So those are like biobanks, EHR, all the phenotypes, you know the meme, right? You just saw me make the hand gesture, it's great. And actually, indeed, I used to have that meme in these kinds of talks. But I think it's also important to think about one very specific illness at the same time, because these general things are general. You're never going to go deep on a question. I mean, I like found the chronotype thing and then did some grubbing around on PubMed, but I'm not an expert on circadian rhythm biology by any stretch of the imagination. It's just what the literature provides. I have, however, been working on schizophrenia genetics for quite a long time. And, schizophrenia as a phenotype is characterized by hallucinations, delusions, flattening of affect, loss of joy in life, inability to organize one's thoughts, oneself, care for oneself, communicate clearly, think and speak in a way that others can follow. Often clinicians will describe speaking to an individual with schizophrenia that they express nights move like thinking, where the conversation jumps around in unexpected ways and it's very difficult to follow. We have some treatments, they're mostly pretty terrible. And they also carry with them quite often a kind of serious set of side effects. And so to address this, the Stanley Center at the Broad Institute was really founded to work on understanding the biological basis of schizophrenia and bipolar disorder and associated psychiatric illnesses in an effort to change the way we diagnose, stratify, treat those kinds of individuals. Can we learn things about biology of these devastating illnesses in a way that we can maybe think about new mechanistic hypotheses for where they come from to create new treatments. So your own TJ Singh was the lead analyst on the schizophrenia exome meta-analysis first wave paper. This was published in Nature, yes, a year and a half ago, something like that. And the overall analysis focused on about 25,000 individuals with schizophrenia and about 100,000 controls. From on here on the right, you've got a principal component plot of the common variants from the coding region. It's not overwhelmingly genetically diverse from a genetic ancestry point of view, but there's some glimmers of hope there, I suppose. And we performed a series of association analyses. Now, the first thing that we looked at was taking so-called constrained genes. So using large surveys of human genetic sequence data, we can identify those genes for which protein truncating variants are not observed, right? So it's the same sort of what is missing from the airplane in terms of where the bullets hit. If you think of your Wald statistics from the 40s, yeah, love me some Wald. So constrained genes are genes for which protein truncating variants just are not observed at the rate you would expect, conditioned on the mutation rate of the gene itself. And in that class of genes, we see a pretty strong enrichment of mutations that are knocking out one of the two copies of those mutations, one of the two copies of those genes in individuals with schizophrenia compared to individuals that do not have schizophrenia. We see some evidence for this so-called missense 1 class of variation, or this like MPC greater than 3. It's another measure of conservation, of evolutionary conservation, but now focused on missense mutations rather than protein truncating variants. And then a whole bunch of other classes of variation where we're not seeing a lot of signal at all. So interesting, encouraging, but perhaps most importantly, we are now identifying clearly associated genes where loss of function mutations, primarily, though not exclusively, depending on the gene, are sufficiently enriched in individuals with schizophrenia compared to those that do not have it, where we can reasonably confidently say that they are major substantial risk factors for developing this illness. Now, I'll draw your attention to the way that we drew this QQ plot, color-coded. So we color-coded it such that things that increase risk are in this orange to red color, and things that decrease risk are in a light blue to dark blue kind of color. And you'll note all of the true associations are all risk increasing. This is simply a statistical truth that if you take a 1% phenotype and compare it against the rest of the population, it's much easier to detect a 10-fold risk increasing factor than a 10-fold protective factor. If you think like it's got 1% in cases, well, if it's a 10-fold risk factor, it'll be 0.1% in the population. If it's a 10-fold protective factor, it'll be pretty close to 1% in the population. And that just gives you a very different power for the association test, just purely on the math, unfortunately. So if we want to find protective things, it really helps if you can draw from both tails of the distribution, but there is no natural low tail of the distribution for schizophrenia. We don't have a notion of a continuous schizophrenia risk or schizophrenia phenotype. The closest that you come to is something called magical ideation, but it is very far away from what the manifestation of schizophrenia is itself. So you can't do high versus low. You do that for cholesterol. So you can definitely find cholesterol lowering effects, like PCSK9 loss of function mutations. Super easy when you can do the tails, but we can't do the tails for something like schizophrenia, which makes our lives harder, but so it goes. So, yeah, please.
Interesting that
Yep.
in the top are genes that were mutated in a heterozygous loss of function, they call severe neurodevelopmental.
Yep.
So many of those are driving the signal.
This is a good question. So there's very well documented sort of intellectual disability consequences to SETD1A mutations. A few of the others are also identified in the Deciphering Developmental Delay program, or even in the Autism Sequencing Consortium efforts, though not all of them. Indeed, GRIA2A and GRIA3 don't seem to have quite that same sort of presentation. I cannot say with confidence whether there is any cognitive consequences beyond schizophrenia, since we don't have any sort of measure of IQ. But you know, it's a mixed bag. It's a blend. There are some that are very clearly overlapping, and then there are some that are seemingly less relevant to intellectual disability as they are to, say, schizophrenia and potentially bipolar disorder.
I'm not saying that I'm not interesting, even the one that
Yeah.
that are pleiotropic are actually very interesting, because if you manifest as a inflection between the course schizophrenia are very different.
I agree with that, and I will also say that there are a number of examples of particularly the histone methyltransferases and demethyltransferases where missense mutations seem to drive intellectual disability and loss of function mutations seem to drive schizophrenia. And it's not impossible to think that there are feedback mechanisms that will increase the expression of those to compensate, and so you don't get quite as severe neurodevelopmental consequences. But when you have a, you know, basically missense mutation that abrogates function, it doesn't know that it's not making enough of the right thing, and then it's not doing the thing that it's meant to be doing. But that is a bit speculative, just to be clear. Okay, so what have we learned about the genetic architecture of schizophrenia? So over here on the left, we've got those strong acting mutations, those big effect genes that I was, you know, talking about a little bit earlier. We've got copy number variants, large-scale deletions and duplications that have been identified through efforts like the Psychiatric Genomics Consortium, but also the work of people like Jonathan Sabat and Mike Wigler over many, many years, including Nerexin-1, which was pretty much a bona fide association, but also has this complex pleiotropic, many different outcomes, including neurodevelopmental disorders such as autism, as well as schizophrenia. And then over here on the right, you've got all the common GWAS loci. And note, again, that all of our discoveries on the rare variant side are all risk increasing because of the power numbers game and how hard it's going to be to find the protective effects should they exist. Whereas the GWAS loci are pretty much equifrequent above and below that odds ratio equals 1 sort of line. And indeed, most GWAS investigation identifies variation across the allele frequency spectrum that is effectively equally likely to be increasing or decreasing risk. Interesting from an evolutionary selective point of view, because these effects are most certainly in genes that are clearly under purifying selection, and part of that might be manifest through, say, schizophrenia or intellectual disability risk. I also want to flag ACAP11. So putative loss of function mutations were 17 to 13 in schema. We also analyzed bipolar disorder. There's something to the tune of 14,000 individuals with bipolar and 14,000 controls. You put it together, you get a p-value of around 10 to the minus 9, maybe even a little bit better. And there are no de novo loss of function mutations for this gene in the deciphering developmental delay program out of the Sanger, led by Matt Pearls and many others. And when you go and ask, what do ACAP11 mutations do in the oracle that is PubMed, you end up with only a handful of papers, most of them in the Journal of Biological Chemistry, which as a human geneticist, I will admit to not reading. But when I ask my biology colleagues and friends, they're like, oh yes, that's a nice journal. You should trust what's in there. And ACAP11 makes this protein called ACAP220. So the protein name, the gene name is different, very irritating, whatever. And ACAP220 is an anchoring protein. And so what that means is it basically acts as like some sort of shepherd to help, GSK3 beta and PKA get together so that PKA, protein kinase A dependent, the protein kinase A can do its phosphorylation business, which is thought to inhibit GSK3 beta. Now, why am I so interested in this? Why did I take this gene? Well, ACAP11 leads to inhibition of GSK3 beta, perhaps through PKA. It's demonstrated in CAH cells, not in neurons. It's not an easy question to answer. If you knock out ACAP11, you would see an increase in the activity of GSK3 beta. Lithium, frontline treatment for bipolar disorder, inhibits GSK3. Among other things. It's a dirty, messy drug, but the thought therapeutic mechanism of action for lithium is through its inhibition of GSK3 to treat at least a subset of individuals with bipolar disorder. And I think this is tantalizing I don't have an explanation about when ACAP11 matters. I don't have an explanation about what cell populations, if it matters all the time, is it reversible? Any of those sorts of things that you might naturally want to ask me. So I'm just preventing you from asking me those questions because I don't have the answer to them. But it does give you this glimmer of hope, this idea that one of the few pharmacological agents that we have that does anything to ease the illness is now pointing to similar biology from one of the strongest genetic risk factors that we've identified to date for bipolar disorder. That's pretty exciting. As a geneticist, I also get asked all the time, enough of this genetics, GWAS has failed, all your genetics has failed, it's all terrible, why do you still want to do it, it's so expensive. Yeah, I'm sure many in this room have heard those sorts of critiques as well. And so we set about to ask the question explicitly of when is enough enough? When should we, what should we aim for? Where should we go with respect to these kinds of endeavors? And there are a few ways you can answer this question. One is let's saturate genetic discovery, so you can do some pretty rudimentary power calculations. And if you want to pick some point, say an odds ratio of 2, I think before GWAS, everyone would be like odds ratio of 2 for a 1% outcome. What do I care? That's nothing. But now, like GWAS is odds ratio of 1.01, 1.005, like a really small effect. So biologists like, yeah, odds ratio of 2, that sounds great. Let's go for it. So pick an odds ratio of 2. You think of a frequency of 10 to the minus 4, 80% power to detect those kinds of effects. If we get to 250,000 individuals with schizophrenia, it'll take 900,000 individuals. We want to move that line down to an odds ratio of 1.5. So those numbers get scary real quick. But maybe odds ratio 2 is not such a bad thing. And we know things have to exist in this part of the distribution, right? We've already found things up above. And if GWAS has taught us anything, it's like once you start, you actually start to find more things, but they always inevitably have slightly smaller effect sizes than that which came before. It's not perfect. Winner's curse randomly scrambles things, particularly when they're similar variants explained or whatever. But fundamentally, this whole space should have genes in it as well. And so by increasing the sample size to 250,000 individuals with schizophrenia, we can make some kind of progress on that. We can also look at the performance of polygenic risk scores in terms of predicting risk of schizophrenia. I mean, in psychiatry, we have no real blood-based measure. Maybe a PRS is that, maybe not. I think that's an open question that I'm sure that the Precision Medicine seminar series has been grappling with over its entire existence. But we can certainly ask the question of how is this working across different genetic ancestries? And what we have in this picture is work from the Psychiatric Genomics Consortium Schizophrenia Work Group showing that the performance in individuals of broadly European genetic ancestries consistently outperforms individuals with broadly East Asian genetic ancestries. And that's pretty much consistent across cohorts, consistent across where you construct your PRs, very active area of research of how to improve those sorts of things. But this is just, simply a reflection of what I think Alicia Martin so clearly articulated in her paper in Nature Genetics in 2019 about how increasing genetic distance from the group of individuals for which you performed your discovery GWAS further weakens the performance of these polygenic risk scores. Finally, I think this is perhaps maybe the easiest to intuit, and yet, at least in my mind, actually the most persuasive argument for trying to get to 250,000 individuals with bipolar disorder, schizophrenia, and that's this question of carrier studies, right? I would like to know whether 200 people that carry ACAP11 mutations respond to lithium. We looked at 7 in the initial discovery. 7 is not enough to tell us whether it's doing any better or worse. It doesn't work that way. We had a couple hundred, maybe we could start to do some like actual science on that kind of question, get to some place of confidence about whether or not there is any specificity to individuals that carry these kinds of mutations as it pertains to certain outcomes versus people with schizophrenia that don't carry those kinds of mutations. And so it's worth just reflecting on what the median profile of these kinds of associations look like. It's about 1 in 1,000 in individuals with schizophrenia or bipolar disorder, about 1 in 10,000 in the general population. And so if I want to do a carrier study amongst individuals with schizophrenia in a very well-powered setting, I'm going to need 250,000 people with each, schizophrenia and bipolar disorder. And so that's what the Stanley Center has set as a goal. Here are our sample projections for how we're going to get there. I love this slide because it's got flags on it that I bet people in this room can't guess. I'm really, really confident that there are flags on this slide that people can't guess. But in any event, we have efforts already sequenced, about 50,000 individuals overall with schizophrenia. There's recruitment efforts underway or samples collected and available at Broad for sequencing to the tune of about 120,000 individuals for schizophrenia, about 100,000 individuals for bipolar disorder, and nearly 160,000 control samples. And for the most part, we have sequenced at the Stanley Center equal numbers of cases and controls. Now, some of these flags, anyone know this one? No one? No. Well, that's Ethiopia. Okay, how about this one? The next one is South Africa. This one's Kenya. How about up here? Yeah, okay, now you've all gone quiet. I do it the other way around, it's a trap. Anyway, yeah, so we got Ireland, UK, Finland, Sweden, Netherlands. I mean, the diversity problem that's present in genomics is present in the work that we've done, certainly. But I think one of the most important aspects of this work is that we've really tried to at least address the question of how does the genetic architecture of schizophrenia look across the world rather than just in a small subset of that world. But we did it not just in a give us your samples and we'll sequence and then write the paper and you can be an author. We did it in a way where we partnered with psychiatrists and researchers at each of the institutions. We had a training arm. So the marquee program is a program called NeuroGap that was led by Karsten Konen. And as part of that effort, we did the recruitment and the science side on this, purple line. But the green line was focused on training and education, building research infrastructure and capacity so that everyone can participate in this research and join together in partnership. Collaborative partnership is really like an important part of the way that we approach science, I think. And we can do a lot to address, longstanding inequities by making these kinds of efforts as part and parcel of our recruitment and analysis efforts. The Ginger program that was down there on the bottom has, got 24 different African research fellows from across the different recruitment sites. You can see all of the statistics of training and workshops and how that all operates. The training is on how to be a scientist, how to write a paper, how to analyze a data set, how to actually contribute to this shared effort that I think we all are on of understanding such a devastating illness like schizophrenia. On the genomic side, we have developed this blended genome exome sequencing technology. So the idea here is that we get somewhere in the neighborhood of 30 to 40x depth sequencing in the exome, couple that with about a 3x genome, low-pass genome, done in a single sequencing run. Actually, it's done in many sequencing runs in a pooled fashion, but still it's like one library that we're sequencing over many lanes. And we've developed, kind of variety of evaluations of how well this kind of approach works. Here's some pictures of the genome coverage on the, kind of low-pass sequencing side. And then on the y-axis, we've got the R-squared of the imputed common genetic variation. And you can see that like when we get to around like 3x, where we tune the technology, you're more or less getting maxing out. There's like, you can keep going, but there's like diminishing returns at that point in time. The GSA, the global screening array from Illumina, gives you this performance. That's invariant because genotype a bunch of sites, then try and impute. And then on the exome side, we're well past the high degree of consistency in the rare variant capture. So those rare coding variants that I talked about delivering actual genes very rapidly, we made sure to capture those, but then do the low pass genome to give us the GWAS component itself. We blended the libraries a bunch of different ways and we've settled on the technology now. And now this is in production at the Broad Institute and piling through those sequence samples that I mentioned earlier. So to close, genetics works. We can find things about any question in biology can be answered in part by genetics. It can help you figure it out, but it's hard. It's really hard, but you can do it. I promise. better phenotypes generate better results. I think that's an important thing to bear in mind because I have spent a fair amount of time talking about what I would describe as potentially very noisy or even, dirty phenotypes, still delivering us something. But the fine scale phenotypic information is, I think, really important for following up and interpreting what those genetic associations are doing, translating the genetic discoveries into biological hypotheses, into understanding of mechanism, all very, very challenging. And I think that's where the rare coding variants in particular offer near-term biological insights, even though they capture a vanishingly small fraction of the overall genetic risk, which is still and remains tied up in common genetic variation. So that I'll say thank you and open the floor for any more questions.
This was an amazing talk, and we do have some online questions. So actually, Sharif was the first one to ask a question. Sharif, feel free to unmute yourself. Should be able to speak.
Hi, Ben. Thanks for the talk. Just a quick question with regards to what you were mentioning about protective effects for phenotypes like schizophrenia. So you mentioned that the power is difficult to identify protective effects, but how would like a study design where you're looking at just at individuals where you predict them to be at a high risk of disease and then identifying individuals who don't go on to develop that severity of disease? How would the power for that work?
Yeah, so, like maybe common genetic background could be used to stratify risk and boost power a little bit. There's sort of two challenges with that. The first is you need good phenotype information on those individuals. And in a lot of instances in the case control setting, we have limited to no phenotypic information on those individuals. And so you'd want to be certain that you aren't, in a sense, making some diagnostic or phenotypic error in that context. But the second is like, the SNP heritability for schizophrenia is moved in the kind of 20 to 30% range. And that only gets you so far down the tail. There's still the selection that you would get therein is still modest. And you might get a little bit more power, but there is a risk of bias. And then any phenotypic misclassification, I think, would really, really cause you a lot of headache and pain in that kind of context.
Thanks. And there's another question from Abby. Abby, please, you can unmute yourself.
Hi, yeah, I just had a question whether the summary statistics are available for the rare variant studies.
Yep, you can go to GeneBAS for all of the UK biobank ones, genebas.org. You can go to schema.broadinstitute.org for all of the schizophrenia ones, and bipex.broadinstitute.org for all of the bipolar association results, as well as a few links to some other disease association result sets, including epilepsy and autism, that you can browse from there.
So I have a question as well. So I really like the blended genome approach and or sequencing approach. And could you comment on the cost and also apart from that, what are the downsides?
Right. So cost is somewhere in the neighborhood of between $100 and $150, depending on sample volume and when you do it. We're evaluating the NovaSeq X as part of the approach, but I think by the end of this year, offering it at about $100 a sample is quite a strong likelihood of reality. I think at volume, there are volume requirements in terms of number of samples that you do to get that, and there'll be some slightly dynamic pricing therein. The upsides are you get the GWAS common variation, you get the exome variation really well called. We can even do somewhat comparable large-scale deletion or duplication calling based on the sequencing information that is akin to, in the same ballpark as the arrays, maybe even slightly outcompetes slightly less dense array in terms of calling the structural variation. You don't get rare non-coding variation terribly much or terribly well. How to analyze and annotate that's kind of harder, which is part of the kind of design consideration. And there are other flavors of structural variation or smaller, particularly smaller, insertions and deletions that would be, that are still larger than you would get in a read for the coding region, but maybe on the order of 1,000 or 2,000 bases that are going to be very, very challenging to call them that compared to something like a deep genome. The trick with the deep genome, though, is that it's like 3 to 4 times more expensive. So, the kind of cost benefit equation, I think, is pretty clear.
But you probably need to sequence controls as well, whereas a lot of deep genome sequence controls are already available.
Yeah, I mean, to a degree, the deep genome sequence controls are available. I think it's challenging to integrate the genome sequence data sets that exist right now and use them as controls in a case control kind of fashion. Not impossible, just challenging. But if I do equal numbers of cases and controls versus just doing cases on something that costs 4 times as much, I still save half the money. And I have a lot of the variation that I care about that is most interpretable for biology and for, say, epidemiology, if you're interested in population risk and modeling and those sorts of things. So it just sort of seems to sit in that convenient, but it's obviously, I mean, in a decade, this technology won't matter anymore. And we hopefully, I would presume, be it, deep sequence on everyone is a kind of relatively standard operating protocol.
Can I follow up on that one?
Sure.
I'm sorry. You use, how did you come up with the threshold of 30-40x? So traditionally, we say 50 to 60 is a basic exome. We argue for 100-150x, oftentimes for more like a clinical exome. And so your capture is much better. And if it's better that much, can you capture also mosaic variants in this settings?
So I'll do the mosaicism first. Mosaicism is hard because the mosaic fraction is critical. And so for low levels of mosaicism, it's just going to look like error. And there's no 2 ways around that. Like if you're only seeing 2 or 3 reads supporting an alternate allele, that's as likely to be a sequencing artifact as it is to be a kind of mosaic variant that is actually propagating or expanding, depending on your, I guess, question and maybe biopsy. In terms of where we settled on the 30x, it was really based on this set of analyses, and we looked at it, in rare variation in particular. So to deliver a, high confidence singleton mutation, the mean coverage is what is easy to describe. The protocol is, I think, something like 80% of the target at maybe 15x, something like that. So we do keep an eye on the distribution of coverage across the target as a readout for the sample quality, because once you're at like a dozen reads for a true biallelic site, you're going to call that pretty confidently almost all of the times that you're going to see it. So you'd have to get very unlucky to miss the one important mutation in that kind of approach. It's not necessarily the clinical setting. Like maybe if you wanted to do the clinical stuff, you would do this twice, but still like $200 less of that, right? Like, I don't know. Tricky. And there are, I mean, like even just the exome itself, target capture does introduce some biases and some error modes, and there are like false variants that do crop up as a consequence of doing the library construction this way. Hard to know where they're coming from, but certainly pretty clearly documented comparing genome sequencing versus exome sequencing. There's a small collection of false mutations induced by that library construction process, but pretty modest. So, and then the QC does a pretty good job of cleaning up.
So the common variants are fortunately shared across many regions. The rarer you go, the more low blood they would tend to be, to some extent.
Well, until you get to the things that are only newly arising every generation. So for the things that have a selection coefficient of 1, they will be completely randomly distributed across the population, because they won't propagate. So there's this sort of, that makes sense?
No, that's
so, but if I carry a variant that neither of my parents carry, then my genetic ancestry matters not one whit.
Yeah, of course.
Yes, so de novo mutations are completely random.
Okay.
And even I would say like things that are like 2 or 3 generations old on average, so things with selection coefficients in the like 0.1 or 0.2 and above range, then geography doesn't matter. There is an in-between space where geography starts to matter in a much more concentrated way, and potentially even a more local and sort of discretized way than the continuous variation that you see in common genetic variation. So it just depends on where you are in the age of the mutation for how much it matters.
If you want. Yeah, go ahead.
In the multi-ethnic GWAS effort, did you find any evidence that ancestry specific genetic structure?
So in the PGC, let's skip going to the side. So in the PGC schizophrenia work, there was an effort to bring together cohorts across East Asia. And, in that GWAS, you have the loci you identify from the cohorts largely recruited from Europe and the US, so broadly European genetic ancestries. And comparing those two data sets, in the event that the variant was common enough to be reliably tested in both groups, they showed highly concordant effects. So the association effects were very similar. The magnitude and direction were largely preserved, even across these very disparate environments and all kinds of things. There are lots of differences potentially popping up, and yet still the common variation is broadly shared such that it is common enough in each of the groups. You get a huge amount of new genetic variation that you test when you are looking at parts of the broader human family tree that we all share that are comparatively understudied because they have, common variants that aren't common anywhere else in the world. Most of the common variants are found somewhere else, but the frequency differences may enable and often do enable much more direct and better powered association testing. And so I think my general impression, at least as it relates to schizophrenia, is that broadly the genetic architecture is not necessarily composed of exactly the same variation, but that variation which is shared contributes similarly, suggesting that the pathogenic processes across these diverse environments are at least shared as it pertains to what we're discovering from this kind of genetic analysis.
Maybe last questions because
Okay.
we're over time. How much heritability do you think is missing by misspecification of the model or using additive model for common variants, for example?
So we looked at adding the second degree of freedom across many continuous traits. That paper just came out in Science, and the contribution of non-additivity within locus is marginal, almost undetectable for most outcomes. There are a few exceptions. It's actually easier to find loci than it is to find the LD score regression signal, even in something like UK biobank. Curious, kind of interesting. And like technical artifacts become a much bigger problem in the game of non-additive association than in the game of additive association models for reasons about non-random genotype fallouts and all that kind of stuff. But most of the detectable risk that we can see is still well characterized by the additivity. That does not foreclose on the possibility that there are epistatic interactions or other sources of variation that you would pick up in twin and family studies that you would have a much harder time accessing in kind of random case control style association studies because of the relative amount of genetic correlation between a pair of relatives versus a pair of random people from the population.
I was most curious about the recessive effects for a disease like schizophrenia, for example.
Yeah, I mean, we look and it doesn't, nothing obvious, but I think there's, but it's also underexplored. So I wouldn't say that nothing obvious means nothing there.
All right, great. Thank you so much.
Of course.